But finally, we're getting started. It looks like we landed on the Mars or something. That's how it felt. But anyway, we are very happy to have Carolyn Klinge talk to us today. Of course, I think 99% of us don't need any introduction to Dr. Klinge. She's professor of biochemistry and molecular genetics. She has worked with us for a long time. She's, an NIH-funded investigator for several decades now and has worked closely with Dr. Winters over the last 15, 20 years, established a good working relationship with endocrinology and helps us run the molecular part of the endocrine grand rounds. And we are very grateful for her to make those arrangements and find speakers for those topics, which we all enjoy. So without much delay, I'll have her start to talk.
Thank you very much. Thank you very much for having me here today. So I get to take off my mask. This is like, amazing to be in this building and have my mask off. So I'm going to talk about an RNA binding protein called hnRNA2B1 and its role in regulation of endocrine sensitivity and resistance in human breast cancer cells. I have nothing to disclose. So what I'm going to talk about today first is Epitranscriptomics, the writers, readers, and erasers of one of those epitranscriptomic marks, which is methyladenosine at the 6th position, N6-methyladenosine, or M6A in RNA transcripts. And then, because this is an endocrine seminar, I thought, well, endocrinologists may wonder what in the world this might have to do with anything associated with medical endocrinology practice. And there may be a role for an M6A reader in linking obesity and diabetes and breast cancer. So that should perk your interest, things to come. Then I'll talk about some of the data that we've collected in my lab characterizing the role of hnRNA2B1, which I will try to refer to as A2B1 because it's such a mouthful otherwise. And then, of course, thinking about future studies, collaborations, suggestions for this research, I'm all ears. Okay, so what's epitranscriptomics? Epitranscriptomics is the covalent chemical modification of transcribed RNA. And these days, pretty much all the genome is transcribed. So this would include messenger RNA, ribosomal RNA, the link RNAs, the primary transcript of microRNAs, so everything that's transcribed. And there's over 160 chemical modifications that are known, and they occur on all the ribose bases that you can see here. Now, the most common one is M6A. So this is M6A. You see the methylation at the 6th position on adenosine. Then the next question is, well, where are these transcripts the epitranscriptomic marks on the transcript? Well, you can see that it will vary across the transcript. Here's the cap, the 5-methyl cap, and then throughout the transcript. M6A is localized to the 3' UTR, but it also exists within the coding sequence and also in introns within the gene. Now, the role of M6A, because it is the most common epitranscriptomic mark in RNA, is a hot topic. And that's shown in this diagram that I made, a chart showing the yearly publications from PubMed from 1996 to the present time. And of course, we're early in 2021, so not that many papers per year. So it's just going up and up. So a lot of interest there. Now, there are writers, readers, and erasers for the M6A mark. So the writers are in this complex with metal 3, and they actually add the methyl group to that 6th position on adenosine. So that's the writers. And this is a removable mark, so there are also erasers, and that's ALKBH and FTO. FTO, of course, does have a role in obesity and diabetes as well, so there's that. And then the role of the M6A mark depends on what readers are reading that mark, which is cell context specific. So here's a couple of readers, and there's that hnRNA2B1. Sometimes you see small letters HN, heterogeneous nuclear, and sometimes capitalized. The actual, I don't know how to say it, correct gene name or protein name would be capitalized because we're talking about a human protein. here. But these readers are involved in processing of primary microRNA, X chromosome inactivation, splicing transcripts, stabilizing transcript, exporting the transcript into the cytoplasm for translation, involved in translation, mRNA decay, and RNA subcellular localization. So all the events that we think about when we think about what happens to transcripts. And this just makes the point that the M6 transcript location and readers will regulate each transcript's fate in a cell context specific manner. Now, one of the readers that was not included, there's actually a number of readers, and one of the readers that wasn't included in the last slide is IGF-2 BPs, and particularly IGF-2 BP2, which for endocrinologists may sound a little bit familiar. And that's because IGF-2 BP2s, SNPs, are common in diabetes, and this results in decreased expression of IGF-2 BP2, which plays a role in diabetic nephropathy. And so here's IGF-2 BP2 acting as an M6A reader and in its role in diabetic nephropathy. But IGF-BP2 is involved in the linking between obesity and diabetes and also to breast cancer. And that's covered in this particular diagram from a review in endocrinology. So you all are very familiar with this looking at this sort of thing. But what happens is that, in fact, there's a decrease in IGF-2 BP2, and that results in an increase in IGF-1, and bad things descend from that. But that also increases breast tumor cell growth. So we can connect not only through estrogens, aromatase, fat-making adipose tissue having increased aromatase expression and increased conversion of adrenal androgens to estrogens in postmenopausal women in particular. So you might wonder, how did I, who am a breast cancer and microRNA kind of person, get interested in M6A? Well, there were two papers published in 2015, and these again are the core proteins that identified hnRNA2B1 as a reader of the M6A mark that was added by metal 3. And that hnRNA2B1 associated with the DROSHA complex within the nucleus and increased the processing of primary microRNA, which is very long, into precursor microRNA, which is shorter and has a stem loop. Now, that's not the mature form of microRNA that is made in the cytoplasm by the dicer complex. That's very short, 22 nucleotides in length. And you can see that these papers are heavily cited. So this is a foundation for a lot of people's work in the field. So what's hnRNA2B1? It's an RNA binding protein. We know that it's involved in reading the M6A mark. It has two RNA recognition motifs. And what's different between A2 and B1? Well, the difference is that the B1 isoform has 12 additional amino acids at the N terminus. So it binds to RNA, and that's depicted in this model. And hnRNA2B1 is also involved in the processing of microRNAs and link RNAs into exosomes or extracellular vesicles for export from cells, and that's depicted in this diagram. In addition, hnRNA2B1 has many roles in the cell in addition to its M6A reader activity. So we know it's involved in splicing, in cleavage, and in transport of transcripts into the cytoplasm. Now, Aaron Johnson, who is at the University of Colorado, did a study in which he performed, or actually grad students in his lab performed ECLIP, enhanced cross-linking immunoprecipitation, to identify the transcripts that were recognized by hnRNA2B1, both hnRNA2B1, both, and hnRNA2B, the B isoform alone. And they found the distribution in MCF7 cells of where hnRNA2b binds, and that's depicted here. And there were slight differences, particularly within the proximal intron between both A2 and B1 and B1 alone. So it binds to RNA in MCF7 cells. So I was interested in finding out what is the role of epitranscriptomics in acquired endocrine resistance in breast cancer. So in my lab, we use a breast cancer cell line model of acquired endocrine resistance that starts with MCF7 cells, which are dependent upon estradiol for their growth, and they are growth inhibited by tamoxifen, an anti-estrogen, a selective estrogen receptor modulator, and they express estrogen receptor alpha. When breast tumor cells progress, they move from tamoxifen sensitivity to tamoxifen resistance. And this is modeled in cells that were obtained from Robert Clark at Georgetown University, LCC9 and LY2, which are progressively more tamoxifen resistant. These cells grow in the presence of tamoxifen or other selective estrogen receptor modulators or the selective estrogen receptor degrader fulvestrant. So they don't depend upon estradiol. So they're endocrine resistant breast cancer cell lines. So I wondered if there was a difference in the expression of any of the writers, readers, or erasers in these different cell lines compared to MCF7 being tamoxifen sensitive. And the answer was the biggest difference I saw was in hnRNA2b1, this reader of the M6A mark. So here is the relative hnRNA2b1 protein expression in MCF7 set to 1 over multiple experiments compared to the two tamoxifen-resistant cell lines, LCC9 and LY2, and we can see higher protein expression of hnRNA2b1. Well, that might be nice, but if it has no bearing on human breast cancer, who cares, right? So you can look in lots of databases and find out how your favorite protein or gene is expressed in breast tumors. And so I did the same thing, and I found higher hnRNA2b1 in breast tumors compared to normal breast. That higher expression of hnRNA2b1 in all breast tumors is associated with a lower probability of relapse-free survival. Oops, back. And that's true in estrogen receptor positive breast tumors as well. If we look at what other people have reported about hnRNA2b1 in breast cancer, we see that these three papers show higher expression of hnRNA2b1 in breast tumors compared to normal breast tissue. But there was a monkey wrench thrown into the mix with the publication late last year of a paper in eBiomedicine by this group of investigators who showed, in fact, that hnRNA2b1 transcript is lower in breast tumors and that low hnRNA2b1 associates with lower overall survival, and that hnRNA2b1 transcript is lower in invasive ductal carcinoma in situ compared to ductal carcinoma, in invasive breast cancer compared to ductal carcinoma in situ. Yeah, human. Yeah. Now, of course, they were mining data, but they also did a TMA, a tumor microarray, and with 50 pairs of breast tumors and their corresponding lymph node metastases, and they found lower protein expression of hnRNA2b1 in the lymph nodes compared to the primary breast tumors. What they don't really compare, well, okay, I'll stop there. So their conclusion in this paper is that that decreased hnRNA2b1 stimulates metastases. So, of course, I dug into a database called HCMDB, the Human Cancer Metastasis Database. And according to that data, the hnRNA2b1 expression is higher in primary tumors with that metastases compared to primary tumors without metastases. And there's higher expression of hnRNA2b1 in, I would call these distant metastases compared to drainage through the lymph node, which, depending on what metastatic person expert that you talk with in the breast cancer field, like Danny Welch, he sort of doesn't think that lymph node metastases are really exactly metastases. They're different rather than because they haven't traveled that far. They haven't gone out into circulation and gone to the brain or the bone or the liver or the lung. And we see higher hnRNA2B1 in the metastases in liver and lung compared to lymph node in this database. So maybe hnRNA2B1 is doing something interesting as it gets out to set up metastases. So I am testing the hypothesis that hnRNA2B1 overexpression stabilizes primary microRNAs that are processed to precursor and mature microRNAs that act on target and pathways to promote endocrine resistance. So I would expect some increases in what I would call breast cancer progression microRNAs. So to test this hypothesis, I was fortunate enough to get an R21 grant funded. And the goal was to overexpress hnRNA2B1 in MCF7 cells and knock it down in LCC9 and LI2 cells. And by overexpressing it in hnRNA2B1 cells, I wondered if I could convert those MCF7 cells that are tamoxifen sensitive into becoming tamoxifen resistant. And conversely, if I knocked it down in LCC9 and LI2, would I restore endocrine sensitivity? The bottom line is yes. But I don't know quite the mechanism. But we have a number of observations that I'm going to share with you. So what I did was to purchase a plasmid expressing human hnRNA2B1, the B1 form, so not just the A2, and then selected for stable overexpression with G418 over time. And this is the stable cell line, overexpressing hnRNA2B1, and we have about a 4.5-fold stable overexpression. So it's a modest stable overexpression. And that we can compare to LCC9 and LY2, the tamoxifen-resistant cells, and the parental MCF7 cells here. But for that's sort of over time. But what I initially did was to do a transient transfection for 48 hours or 72 hours and perform microRNA RNA-seq. And these data, I did 5 replicates of each control versus transfected with hnRNA2B1 at the 48 and 72 hour time points. So those are the data, and they were analyzed by Eric Rushka. And so I'm going to put it this way. I then performed core enrichment pathway analysis of all the differentially expressed microRNAs, both at 48 hours and 72 hours, looking at those that were up and down regulated at each time point in the pathways associated with those changes in microRNA expression. And I've put in pink boxes those that are related to breast cancer. We confirmed by quantitative real-time PCR, and this is work from Kelly Peel, who works in my lab, overexpression of 3 selected microRNAs, MIR 1266 5P, MIR 1268A, and MIR 671 3P. And these are microRNAs that are known to be overexpressed in breast cancer and play some role in breast cancer. Well, that confirmed part of my hypothesis that we would see an increase in microRNAs if we transfected MCF7 cells with hnRNA2B1, and that these microRNAs would have roles in processes that would contribute to breast cancer progression. But why are we seeing downregulation of microRNAs? If hnRNA2B1 is doing something to upregulate primary to precursor microRNA biogenesis, why are we seeing downregulation? Well, because hnRNA2B1 can interact with positive or negative regulators, so it can also decrease the expression of specific microRNAs. So if we look at the microRNAs that are decreased upon hnRNA2B1 overexpression and focus in on those pathways, we see some of the same pathways, TGF-beta. We also see microRNA-dependent EMT, activation of Notch, and other pathways. And we confirmed that 3 of the hnRNA2B1 regulated microRNAs were in fact downregulated in the stable hnRNA2B1 overexpressing cells, and that's shown here. miR29A3P, miR29B3P, these are both associated with decreased overall survival in breast cancer patients. And miR2223P is downregulated by hnRNA2B1, and it targets estrogen receptor alpha. Now, Eric Ruska performed in silico identification of the mRNA targets for all the microRNAs that we identified using miR-TAR base, and then looked for validated gene targets in category compare. And then performed gene ontology biological process enrichment for the genes targeted by the differentially expressed microRNAs. And so again, we come up with the TGF-beta pathway. Also, the cell adherence junction assembly, stem cell population maintenance, response to estrogen and estradiol, positive regulation of protein serine threonine kinase activity, and many others that you can see here. Serine family amino acid metabolic processes, response to steroid hormones, androgen receptor signaling, and mesenchymal transition, EMT. So, first summary here of what we've learned from the myrome that we just performed is that overall we saw both up and down regulated microRNAs regulated by hnRNA2B1 overexpression, and using Metacore pathway and gene ontology biological processes analyses, the results support a tumor progression role for the microRNAs that were regulated by hnRNA2B1 overexpression. Well, I mentioned that we have created a stable MCF7A2B cell line over time, and the cells are shown here. And so these are photographs that were taken by Belinda Petrie with thanks to Matthew Nystoriak using his microscope up on the second floor here in the Delia Baxter building. One of the things that I've always seen with the MCF7A2B1 cells is they grow like pyramids. MCF7, they're flat, they grow fluid monoliths. Not the MCF7A2B1. They like to grow as pyramids and then float away. So that's an interesting observation, and I have yet to fully determine what that means. But I did ask Dr. Michael Heckman to look at this because he's an expert at cells, and he said the cell aggregates are, at least in his opinion, having to do with being stem cells, which fits the possibility that I had initially put into the grant, which is that overexpression of hnRNA2B1 would increase cancer stem cell biogenesis in the cell. So that's a pathway that we need to go down. And we've started that. Belinda has started working on that. So the question we asked, so this is all unpublished stuff now, by the way, starting with here, but none of this is published. This is all new. Does stable hnRNA2B1 overexpression, about 4.5-fold, alter the response of MCF7 cells to estrogen receptor alpha antagonists, namely 4-hydroxytamoxifen, the active metabolite of tamoxifen, and fulvestrant? And so this is an MTT assay, and these assays were performed by Belinda Petrie in the lab. And what we're looking at is MCF7 cells, and they are growth inhibited by 4-hydroxytamoxifen, but in purple, the MCF7A2B1 cells are not growth inhibited by tamoxifen, nor are they growth inhibited by fulvestrant. And that makes them similar to LCC9 cells, the tamoxifen-resistant cells. So this suggests a role for the overexpression of A2B1 in endocrine resistance. Now, if that's true, I should be able to knock down hnRNA2B1 and restore endocrine sensitivity in the MCF7A2B1 cells. So I transfected MCF7A2B1 cells with siRNA to A2B1, and Southwit, who was a medical student in my lab not last summer because there was nobody there in the lab last summer other than me and Belinda. The summer before actually did these MTT assays. And what we can see again is that there's the MCF7A2B1 cells that are not responding to by growth inhibition to 4-hydroxytamoxifen or fulvestrant. But when you knock down A2B1 expression in these cells, I forgot to put in the Western blot to show that I in fact knocked it down. But nonetheless, what we can see is that we now restore endocrine sensitivity, at least to high concentrations of 4-OHT and to 100 nanomolar fulvestrant. So this confirms a role for the overexpression of A2B1 in endocrine resistance in MCF7 cells. So if that's true for the MCF7A2B1 cells that I've forced to overexpress A2B1, would it be true in the LCC9 and LY2 tamoxifen endogenously tamoxifen resistant cells that of their own overexpress A2B1? And so here I've put in the Western blots to show that we got knockdown of A2B1 when I transfected the cells with siHNRNA2B1, and that's shown graphically in panel C here. And then we have MTT assays both in LCC9 and LY2 cells. So the control transfected cells are not, neither LCC9 nor LY2 are not growth inhibited by 4-hydroxytamoxifen or fulvestrant. However, when the cells are transfected with the siA2B1, we now see a gain of tamoxifen and fulvestrant sensitivity in the cells. So this confirms a role for A2B1 in endocrine resistance in LCC9 and LY2 cells. Now, in thinking about hallmarks of endocrine resistance, I wanted to know if MCF7A2B1 cells show enhanced migration and invasion, which are properties of endocrine resistant cells. So Kelly Peel ran Boyden-Chamber assays in the lab, and these are migration assays, and were okay. This is migration, I think. Well, I'm not sure right now. Oh yeah, migration. Yep, these are migration assays, and what you can see is that overexpression of A2B1 in MCF7 cells mimics the enhanced migration that we see in the LCC9 and LY2, and increases, this is cell invasion, increases the invasion of the cells similar to that which we see in LCC9 and LY2 cells. So this is a gain of function in terms of enhanced migration and invasion. So again, if we knock down A2B1, will that inhibit the migration of the cells? And the answer is yes. And so I transfected the cells, and Kelly ran the Boyden-Chamber assay, so we have migration on the left and invasion on the right. And what we can see is that if we transfect with siA2B1, we inhibit the migration of LCC9 and the MCF7A2B1 cells, and likewise inhibit their invasion. Again, suggesting a role of A2B1 in cell in migration and invasion. Now, clonogenic assays are used to examine the ability of a single cell to grow into a colony. So what you do is you seed a small number of cells into wells and then let them grow over a long period of time. So this is 14 days of growth, and you just stain them with dye and then use image J to analyze how many colonies have formed, and you can also measure the colony size using image J. So I asked the question, does siA2B1 inhibit MCF7A2B1 and LCC9 clonogenicity? And what we can see is if we knock down, so this is colony forming units basically, and what we can see is that we get, if we knock down A2B1, we see a reduction in clonogenicity of LCC9 and MCF7A2B1. cells, and that the size of colonies is also decreased when we knock down A2B1 in both LCC9 and MCF7A2B1 cells. So this confirms a role for A2B1 in clonogenicity in MCF7A2B1 and LCC9 cells. Now, soft agar colony formation assays are used to evaluate anchorage-independent growth ability of cells, and that's a hallmark of tumorigenesis and metastatic potential. So I asked the question, does A2B1 enhance colony formation of MCF7 cells? So we're comparing MCF7 cells and MCF7A2B1 cells. And right away, I mean, it's very visual, right? MCF7A2B1 have higher increased colony size compared to the parental MCF7 cells, and that's seen in this chart over here. But the colony number doesn't really change. So again, if we knock down A2B1, will that inhibit colony formation? And what we can see is that we now reduce the number of colonies formed for both LCC9 and the MCF7A2B1 cells with knockdown of A2B1, and that's the red lines shown on the left. And but the colony size doesn't change that much. We do see a decrease at 7 days in the MCF7A2B1 cells transfected with SiA2B1, but not at the end. So there's a variety of potential explanations here, such that, well, you're having a diminishing return because you transfect the cells with X molecules of SiA2B1, and then the cells are dividing, they're losing that amount of SiA2B1 that would be present in them. So I'm not sure. So second summary, in terms of the phenotypes of the hnRNA2B1 overexpressing MCF7 cells. Well, what does stable overexpression do? It alters cell growth patterns to form cell aggregates versus a monolayer, and whether that's really cancer stem cells remains to be seen. Stable overexpression blocks inhibition of cell viability by 4-OHT and fulvestrant, so this is a gain of endocrine resistance. It increases cell migration, invasion, colony formation, and these are all hallmarks of the metastatic phenotype. So it appears that hnRNA2B1 would increase metastasis. Conversely, knockdown of A2B1 in the MCF7A2B1 stable cell line in LCC9 and LY2 tamoxifen endocrine-resistant cells restores sensitivity to tamoxifen and fulvestrant and increases migration, decreases cell migration, invasion, and colony formation. So this suggests that upregulation of A2B1 may play a role in endocrine resistance in breast cancer. Now, the next part of the talk, I'm going to look at some of the pathways that were identified in the microRNA targets in the RNA-seq and their association with hallmarks of endocrine resistance in breast cancer. So one of the ones was the regulation of serine and threonine kinase activity. And so what I'm doing here is looking at the phospho-AKT to AKT ratio in LCC9, MCF7, and MCF7A2B1 cells. And what we can see is we have activation of AKT, PI3 kinase, in the tamoxifen-resistant LCC9 and the endocrine-resistant MCF7A2B1 cells. So then, of course, I asked if we knock down A2B1, will we see a decrease in the activation of AKT? And in fact, we do, and that is shown here, both for the MCF7A2B1 cell line and the LCC9 cell line. And you may wonder, why it was so dark here versus here. It's just one of those experimental things. I don't know. Repeat. Okay. Then we also saw activation of MAP kinase in among the pathways. And so we looked at phospho-MAP kinase, ERK1, 2, and MAP kinase in MCF7 cells and LCC9 cells, SI-controlled transfected, SI-controlled transfected, SI-A2B1 transfected. Very visual result again. What we know is that MCF7 doesn't have activated MAP kinase signaling, but a hallmark of endocrine resistance is activation of PI3 kinase and MAP kinase. And so, in fact, that's what we see in the MCF7A2B1 and LCC9 cells, and we see a reduction in MAP kinase activation when we knock down A2B1 expression in the cells. Now, activation of MAP kinase would be expected to increase estrogen receptor alpha phosphorylation on serine 118 in the N-terminus, and that actually can activate estrogen receptor in the absence of ligand, and that's a possible mechanism involved in endocrine resistance, because now you have estrogen receptor active by phosphorylation on serine 118 so that it can bind to DNA and upregulate target genes involved in cell proliferation and anti-apoptosis. Well, and in fact, we do see ER alpha phosphorylation on serine 118 in MCF7A2B1 and LCC9 cells, and we see a reduction in that phosphorylation when we knock down A2B1. Well, how HNRNA2B1 is actually doing this remains to be determined. But Kelly did an experiment, an MTT assay. Oh, no, sorry. This is a soft agar colony growth assay looking at the impact of PD98059, which is an inhibitor of MEK1, and wortmannin, and that's an inhibitor of PI3 kinase activation, and the effect on MCF7A2B1 soft agar colony growth. And what we can see is both PD98059 and wortmannin inhibit colony formation of the MCF7A2B1 cells. We also saw EMT and also cancer stem cells among the pathways implicated as microRNA targeted pathways in the myrome of the MCF7 HNRNA2B1 transfected cells. So Belinda ran a gel looking at E-cadherin and vimentin. A decrease in E-cadherin and an increase in vimentin are hallmarks of EMT, epithelial mesenchymal transformation in cancer. And what we can see is we see a decrease in E-cadherin and an increase in vimentin in the MCF7 A2B1 cells. Neither MCF7 nor LCC9 have undergone EMT, and that's published information. Now, in terms of EMT, the next step, another step in EMT, is that the cells can move to form cancer stem cells. And this is involved in resistance to many drugs in breast cancer, including endocrine therapies. So, Belinda, this is hot off the computer. Belinda is still doing these experiments and working on this. But cancer stem cell biogenesis is measured by the population of CD44 positive, CD24 negative, or low cells by FACS. So Belinda has worked with Dr. Marcin Wozinski upstairs in Delia Baxter. He's in the Department of Medicine and Cardiology, to look at the CD44 positive, CD24 low population. And you can see that she has graphed the data here in terms of the percent CD44 plus, CD24 minus or low cells. MCF7, there's just no cells in that quadrant. A2B1, lots of cells in that quadrant, and this is just in fact an image that Belinda captured showing that number of cells in that quadrant. And we also see CD44 positive, CD24 low, in LCC9 and not that many in LY2 cells. So this is one experiment. She's doing more experiments of that, but this is the latest, and I think it's very exciting because that would fit this model of why MCF7A2B1 cells are growing in these pyramids and then floating off. That would be cancer stem cell formation. So maybe hnRNA2B1 is really involved in cancer stem cell biogenesis. We also saw that response to estrogen, response to estradiol, and hormone receptor signaling were pathways in the microRNA myrome data. And so we looked at estrogen receptor alpha expression in comparing MCF7 and MCF7A2B1 cells, and surprise, surprise, it's actually upregulated. And if you knock down A2B1 in MCF7A2B1 cells, we see a reduction in estrogen receptor alpha expression. And likewise, if you transfect either LCC9 or LY2 cells with siHNRNA2B1, you see a decrease in estrogen receptor alpha. And so that means that hnRNA2B1 is regulating estrogen receptor alpha in these three endocrine-resistant cell lines. Now you might think, well, okay, in breast cancer, aren't you getting a decrease in estrogen receptor so that the cells are now resistant to the drugs that target estrogen receptor like tamoxifen and fulvestrant? Well, it's yes and no. And in some cases, including these cell lines, you're actually getting an increase in estrogen receptor alpha. And so I think it's like, okay, the cell's trying to compensate, sort of like, at first with diabetic patients, right, their beta cells make more insulin and then they're exhausted and they can't make any more insulin. So I think it's the same sort of, Barbara?
So how's that related to the activation or the possible relation of PR alpha?
I think it's a both and, right? So you're getting more estrogen receptor expression and more estrogen receptor alpha phosphorylation with hnRNA2B1 overexpression. Yeah. And so one could ask, if we knock down ER alpha, will we increase endocrine sensitivity? And so this is, in fact, with LCC9 cells. And if you knock down estrogen receptor alpha, you do get an increase in sensitivity to fulvestrant. And this is work from Robert Clark, and Robert Clark is the guru of the endocrine-resistant field in terms of molecular mechanisms, and he made the LCC9 cells in his lab. Okay. So another pathway was identified was androgen receptor signaling. And androgen receptor in breast cancer is an interesting topic that there's divided literature on whether overexpression of androgen receptor is a good marker or a bad marker in breast cancer. But what we found is that, in fact, we see an upregulation. Here's MCF7 cells. Here's LCC9 cells. They're known to have increased androgen receptor protein expression. And here are MCF7A2B1 cells. Likewise, they have an increase in androgen receptor expression. And if you knock down A2B1, you see a reduction in androgen receptor expression in the cell lines. So whether that is a mechanism in endocrine resistance, I'm not sure at this point. We haven't done too much. But Kelly did an MTT assay in which we looked. So there was a recent paper that showed that breast cancer cells that express androgen receptor and are treated with DHT will be growth inhibited. So, you would think if androgen receptor is a bad thing, DHT might increase cell proliferation. So she did an MTT assay with DHT, and what we can see is that, in fact, DHT inhibits the growth of MCF7 LCC9 cells and has less of a growth inhibitory effect in the MCF7A2B1 cells. But since the androgen receptor expression levels are pretty similar between the cell lines, I'm not sure. And in fact, in MCF7 cells, there's hardly any androgen receptor there at all, and yet DHT inhibits. So I don't know what's going on there. So that's why I have the why here. I'll be happy to take any suggestions. So summary 3, we've confirmed pathways in A2B1 regulated microRNA-seq analysis that hnRNA2B1 activates PI3 kinase, AKT, and MAP kinase, and increases phosphorylation of estrogen receptor alpha on serine 1Y8 that activates AR, and increases estrogen receptor alpha and androgen receptor expression, all hallmarks of endocrine resistance. So we have created this MCF7 A2B1 stable overexpressing cell line, modest overexpression of A2B1, and it has the hallmarks of endocrine resistance. So here's what we have so far. We've seen that hnRNA2B1 increases estrogen receptor alpha expression, and that would affect gene transcription. We see an increase in phospho-MAP kinase and phospho-AKT. These target estrogen receptor. I didn't put that in. Oh, yeah, there it is. Target estrogen receptor, so we get increased estrogen receptor alpha phosphorylation. AKT is known to increase androgen receptor expression, so that might be the mechanism involved in that. Activation of PI3 kinase AKT signaling increases vimentin. We saw an increase in vimentin. All these are hallmarks that fit into the tamoxifen-resistant, endocrine-resistant phenotype in the cells. So, thank you for your attention. This work was supported by an R21 grant that I have. Southwit was supported by the T35 NIDDK training grant that we're looking for projects on for this summer. Belinda was supported by a fellowship from the NIEHS T32 grant to David Hine. In terms of the immunohistochemistry on a TMA array that we're doing now, Belinda's working with Brian Clem and Dr. Norman Lehman to read the TMA. And Belinda has worked with Martian and Matt to do FACS of cancer stem cells and image the cells. So that's about it. So thank you for your attention. Oh, and I've got to put up there, today's event code. Barbara?
Do you think A2B working through the microRNAs? And I think in an earlier slide, showed the link between, I can't remember, between microRNA and ER. Right. So what microRNA is that?
MIR222. Well, I didn't put that into this story, but yes. So MIR222 goes. Oh, so go over here.
Tell them the question and then.
Oh, okay, sure. So the question was, so do you think these effects that you're measuring are mediated by microRNA? And I think in part, yes, and in part, no. So one of the microRNAs that we saw downregulated and we had confirmed was MIR222 that targets estrogen receptor alpha. And so we show that it was downregulated, and we can show that we published previously that MIR222 targets estrogen receptor alpha. However, when we knock down A2B1 expression in the MCF7 A2B1 overexpressing cells, MIR222 goes down further. So if it were a direct target, MIR222 should go up. So it's one of those things. I think hnRNA2B1 does many things, not just in terms of being a reader of M6A, but in terms of splicing, transcript stabilization. So, yeah, I kind of feel at a loss to pin down a mechanism, which is what I'm trying to do so that we can get our paper accepted by Cancer Letters. Oh, stop sharing. I'll be glad to do that. Just do that.
Be able to see if they have any questions on the chat.
Okay.
Let's see if they have questions.
Oh, take up. Yeah. So the question is, have you attempted to extend your work on hnRNA2B1 overexpression phenotypes in M-susceptible breast cancer biopsy tissues or primary? No, I have not done that. That would be very interesting to do. I would like to do that. I did purchase a TMA, a tissue microarray, and Belinda stained that with Dr. Brian Clem, and now Dr. Lehman is reading that slide with Belinda. And what we know so far is in agreement with the 3 papers, but in disagreement with the one paper that I said threw a monkey wrench into everybody else's data on H and RNA2B1, is that we see higher expression of H and RNA2B1 in breast tumors compared to normal breast.
For the papers that are using the same database, right, to generate that data? Or did they use their own data?
Yeah, so no, that's another point. So Dr. Clark's point was that, in fact, these people are all using different databases. So you can probably sort through the database and pick the study that will show what you hope to find. And that's a scary thought, isn't it? I've always thought that it would be I have not had a sabbatical, and I'm probably not going to be taking a sabbatical, but it would be fun to do a sabbatical and learn to use databases to mine data and learn things about genes and proteins and their expression. All right. Thanks for coming.
A lot of work there.